Okay, so we are going to get started now. Thank you everyone for joining this webinar, the BGC Argo webinar series. This is hosted by GO-BGC, the Global Ocean Biogeochemistry Array, supported by NSF. It's an infrastructure proposal, and US OCB. They do all of the logistics and hosting for all this, so, big thanks to them. And today there are two speakers, Ellen Park. She is at Hui, and Adam Stower, University of Dalhousie, and I will introduce them in a little bit more detail in a couple of minutes, but those are our two great speakers today. And so before I hand off the mic to them, I just want to go through the usual kind of background and updates of for GO-BGC and BGC Argo. So BGC Argo is a component of One Argo, which is the next phase of the Argo program, the highly successful Argo program. The planning document calls for a total of 4,700 floats. That is consisted of 2,500 core floats. So these are floats that go down to 2,000 meters, measure temperature, salinity, and pressure. 1,200 deep floats and 1,000 BGC floats. And the justification for the size of these arrays can be found in these references down here. And then so one, in addition to adding deep and BGC floats, the One Argo vision aims to increase the density of, Argo floats in these, more dynamic areas of the ocean where we need higher spatial resolution data to properly constrain those processes. And then, so this is the Biogeochemical Argo Science and Implementation Plan. The size of the 1,000 BGC float array was determined through a variety of different approaches, OCBs and decorrelation length scale analyses. And so this was a number that was chosen to constrain, nutrient distribution, oxygen, distribution of CO2 fluxes and inventories. As Argo, as true for all Argo data, BGC Argo data is freely available in real time or near real time. Our goal is to get it online within 24 hours since the float surfaces. There are multiple ways to access the data, but the main data portal that we suggest people access data is the Global Argo Data Assembly Center, and use for BGC Argo data, use the SPROF, which is a synthetic profile. So this is a user-friendly file where all of the BGC sensor data is interpolated onto a single pressure axis. So before there were separate pressures for each BGC parameter, and now it's on a single pressure axis, so it's much easier to use. There are toolboxes that were developed, led by PMEL, so Hartmut and John Sharp. And so this is the One Argo toolbox and written in MATLAB and R, and Yibin. Sorry, I forgot to add Yibin's name to this. My apologies. For the R. And so this allows you to query the GDAC and download data for floats that exist in a certain space or time range, parameters, and things like that. In addition to that, we create 2 data snapshots per year for all the floats that are involved in the SOCOM and GO-BGC program. So these are all data that the data team at MBARI quality controls and creates snapshots. And these can be found in the UCSD library, the digital library. And in addition to the QC dataset, we also produce derived carbon parameters, so PCO2, DIC, alkalinity, and POC, particulate organic carbon, as kind of a value added to this product. And if we have a data FAQ page on our GO-BGC website. And so if you have questions on data access toolboxes, what the QF flags means and stuff, this is a good place to start. The Argo website also has an excellent data access webpage as well. So to get started, I highly recommend reading through all of the documents that are online. So this is what a target BGC Argo array of 1,000 floats distributed globally looks like. This is a hypothetical randomly placed 1,000 floats. So each little white tick is a BGC float, and we are calling for the BGC implementation plan calls for 1,000 floats that have at least 4 parameters of these of the 6 that are currently available. So oxygen, nitrate, pH, chlorophyll A fluorescence, backscatter, which is a proxy for particulate organic carbon, and downwelling irradiance. And this is our current status of BGC Argo. So we're actually, doing pretty well. There are 584 operational floats. Many of them only carry a subset of these available parameters, but 403 of them have 4+ sensors, or it should be 4+ parameters. And that's 62 more than the last time we did this webinar. So that's, so the array is growing, and there are actually now 48 with all 6 parameters, which is really exciting. For what GO-BGC has done, we have deployed 143 floats so far in the program. That's 29 more than the last time we did this webinar. And these squares kind of highlight around the locations where new floats went in the past 6 months or so. We also have 81 float deployments planned where, cruises and floats have been identified, built. And so over the next 6 months or so, these are all the locations where floats are scheduled to be deployed. So you can see, we are starting to fill in the Indian Ocean pretty well. There's going to be I-8 and I-9 cruises down here. And I should mention this is a mix of SOCOM and GO-BGC floats. And then the 813.5 line here will fill in the South Atlantic nicely. And then Kaharoa is going to do this big loop around the Pacific to try to fill in the holes. But one thing that you can see in the array is that there's a hole in the South Pacific here. This is a really hard place to get to. So if anyone knows of any cruise opportunities to be able to deploy floats there, please contact us. That would be extremely, extremely helpful. So in addition to this, there are about 250 BGC floats over the next 5 years scheduled from international partners. These are, kind of plans. Not all funding has been secured for these. And so this is around the locations where the international partners shown on the right are scheduled to deploy floats. Okay, so that's, and then a couple updates on our program. So I think many of you know there was a issue with the Honeywell ISPETs going out of production, but now they are in production. But kind of in response to that, we have been exploring and developing a new ISPET-based pH sensor. So we use an ISPET from Lyonics International. And so it is called the SURF sensor from the version that we built at Embari. So this is a picture of the ISPET on the bottom right and a fully built up sensor on the middle right here. And so we tested the sensor on the spray glider in Monterey Bay, making ascent and descent profiles to about 1,000 meters. And then so the pH profile is shown on the left. There's no noticeable hysteresis going up and down. So that's really good. And the plot on the right shows the measured pH minus an algorithm estimated pH that we know is accurate in Monterey Bay, below about 200-300 meters. So you can see that there's very good agreement between the sensor and the algorithm. So this is, we just did this deployment in December, and we are further refining our design and continuing to test them on floats and gliders. And so the initial experiment was very encouraging, and just big shout out to the sensor team at Embari who made it happen. We received these chips in June or July or something like that. So all this happened within 6 months, which is quite fast. I also wanted to mention that the Argo steering team meeting is coming up. It is March 18th to 22nd. It's in Southampton, but there is a virtual component. It is open to everyone except for one closed session, I believe. And then to participate, you can register at the link using this QR code, or just search Argo steering team meeting 25. And Google will take you there. But if you're interested in, for example, where international partners are scheduled to deploy floats, the Deep and Argo, Deep and BGC Argo programs, statuses and sensor challenges and stuff is a really good way to get background information about the program itself. So if you're interested in that, I highly recommend you listening in to this meeting. You will learn a lot. And finally, GO-BGC hosted a BGC Argo data workshop with support from OCB as well. And so this was a hands-on workshop focused on BGC Argo data access and analysis. So this is a picture of all the participants. It was a 2.5-day workshop, I think, in UMass Boston. So everyone had, got hands-on experience to access the data. And then there were team leads who organized different projects that people can then pursue using BGC Argo data that they learned how to access on day one. So some of the science talks from this workshop is archived on YouTube, but today And this is the team leads, big thanks to them. It was a lot of effort on their part. It wouldn't have been possible without them. And it turns out that the two speakers today were two team leads from that workshop, and then so I'm really excited to hear their most latest and greatest work. So the two speakers today, first talk will be by Ellen Park. She is a fourth year graduate student at the MIT Hui Joint Program. She advised by Rue Nicholson, and then so her research focuses on using the global BGC Argo array to better understand the biological carbon pump. She also participated in the A16 North cruise to deploy a bunch of BGC floats, and she wrote a blog during that cruise too. So that is on our GO-BGC website. And Adam Stower, he is currently a research associate at Dalhousie University, working under Katja Fennell. And so he received his master's last year, 2023. Congratulations, Adam. And his research primarily revolves around phytoplankton ecology and the advancement of optical measurements, especially using those collected by autonomous underwater vehicles like floats and gliders. So two excellent talks coming up. And then so I will pass it to Ellen now. And I need to stop sharing, I believe. Here we go.
Everyone hear me okay?
All right. Yes, we hear you.
Great. Thank you, Yui, for the introduction. And I'm excited to talk to you all today about quantifying biological carbon pump parameters using the Global Biogeochemical Argo Float Array. So as many of you probably already know, the ocean plays an important role in regulating the Earth's climate. It's a large sink for carbon, and since the beginning of the industrial era, it has taken up approximately 30% of anthropogenic carbon dioxide emissions. This uptake occurs due to a combination of physical, chemical, and biological processes, which are commonly referred to as carbon pumps. And today we'll be focusing specifically on the biological carbon pump. The way that the biological carbon pump works is through the sinking of particles or organic matter that are produced in the surface ocean and can reach a depth where they're effectively sequestered away from the surface ocean for a period of time. This pump removes approximately 10 petagrams of carbon per year, which, just to give that some context, is approximately equivalent to annual anthropogenic carbon dioxide emissions. However, there are large uncertainties in the magnitude and variability of the biological carbon pump due to a limited number of measurements across both space and time, and the fact that the ocean is very large and very dynamic, so one measurement is not representative of the entire ocean. When we're thinking about carbon and the carbon cycle, we're often thinking about it in the context of climate change, and so when we're modeling the biological carbon pump, it's been challenging to do this because there's large uncertainties in estimating the transfer efficiencies. The transfer efficiency is a fraction of carbon exported from the surface ocean that makes it to depth, and we can use the Martin curve to estimate these values. So you can see the equation below, and this is derived from an empirical power law fit of particulate organic carbon fluxes. And so what it looks like with depth, there's some schematics of sample carbon profiles that have the same amount of carbon in the surface under two different scenarios. The one in red is when the power law value, or B, or attenuation, is larger, where we have more remineralization and with depth and lower transfer efficiency. We also have the case in blue where the B value is smaller and we have less remineralization and therefore a higher transfer efficiency. So once we know this B value, we can estimate the transfer efficiency using the ratio between two depths. And so when we're thinking often about carbon 2, there's this big question of what we should use as a reference depth. And for the Martin curve, it was typically done with 100 meters, but here we'll be using the primary production zone, which is defined as the depth where chlorophyll A fluorescence is 10% of its maximum. And we'll be evaluating the transfer efficiency at 500 meters. Typically, we measure the biological carbon pump using sediment traps where we're collecting the sinking particles at various steps in the water column, but we can also use BGC Argo floats equipped with bio-optics to see export, or these sinking particles. And this has been done a number of times with several floats when they've been sampling on a higher frequency, temporal frequency than the typical 10-day cycle. And the particular bio-optic parameter that I'll be talking about today is particulate backscatter, which is essentially a measure of how much stuff is in the water. And this signal depends on particle concentration, size, shape, and composition. And as we mentioned before, it can be used as a proxy for particulate organic carbon. So the goal of this project is to quantify the large and small particle transfer efficiencies and compare them between oceanographic biomes using the global float array. So I'll quickly go over the methods. First, all the backscatter data were quality controlled using the EuroArgo real-time quality control toolbox, which is available on GitHub. Then these backscatter signals were decomposed into the large and small particle components. And the way that we did this was following a modified version of the methods from Briggs et al. 2020, where you take the original signal, which you can see on the far left, and you can break it down into a large particle component, which is more of these spiky outliers, a small particle component, which is the smooth profile, and then a refractory signal, which is more of like the background signal, and then a blank, which represents instrument noise. I just wanted to note for all the backscatter people that as of right now, there's been no animal spike removal, but that will be integrated in the future. Finally, the profiles are then assigned to a Fay and McKinley biome. And on land, we can group regions of terrestrial plants that are similar based off of the temperature and precipitation that they experience there. And so we can kind of take a similar approach with the ocean. And this was done by Fay and McKinley using sea ice, temperature, chlorophyll, and mixed layer depth. And the original product was created from 1998 to 2010, which kind of precedes the BGC Argo era. So here I recreated these biomes, these yearly biomes from 2010 until 2023. And you can see all 17 different biomes on the map on the right. And the reason that we want to use this approach is, as opposed to looking at individual floats and saying they're representative of an entire region, we want to group float profiles that fall within similar biological communities that probably have similar carbon export processes, so we can use all of the floats and kind of increase our statistical power. So I'm not going to go into much detail about what all of these different biomes mean, but just to get everyone oriented, each ocean basin is given a specific color, and then within each basin, the darker shades represent the higher latitudes and the lighter shades represent the lower latitudes. So once we've assigned every profile to a biome, we can calculate the transfer efficiencies. And so as I mentioned before, what we're going to do here is fit individual profiles to the Martin curve to get that B value and then calculate the transfer efficiency. So for the small particles, we're fitting the small particle backscatter signal. And on the left part of this figure, you can see all of the small particle backscatter profiles from one float with the solid black line as the median profile values. And so you can see that the median profile gives that nice power law curve. So that works out pretty well. And then for the large particles, we're not particularly interested in the magnitude of the backscatter signal in this case. And if you remember from the previous figure, there's not many spikes, and so we can't fit, it's hard to fit a curve to that. So instead, we're going to look at the spike particle probability, which we're defining here as the number of spikes over the total number of measurements in a given depth bin, which we're using 50 meters here. And you can see on the right, those are the bin spike probabilities from all the profiles in one float, and the median gives that same power law curve as well. And so now we can look at some of the results. And before showing you what this looks like for the all the ocean and all the biomes, we're going to break it down one biome first. So here we're looking at the North Atlantic subpolar seasonally stratified, which is in red and highlighted in this black box in the upper right-hand corner. So here we have on the left the small particles, and on the right, the large particles. The background is a histogram of all of the measurements in the biome, binned by depth, with 1 meter depth bins for the small particles and 50-meter depth bins for the large particles. The solid line is the median value in all of the depth bins, and the dashed or dotted line is the Martin curve using the median of the fit values, so the B values from all of the profiles. And you can see that the median fit recreates the median of the data quite nicely for both the small and the large particles. And you can see too from these figures how we're able to get what the climatological average profile for small and large particles in each biome will look like. So now we can go ahead and look at what these profiles look like across, in this case, 16 different biomes where floats were present. And it's kind of a lot to take in a shorter talk, but you can see that there are definitely differences between biomes where we have variations in the magnitude of the small particle. Sorry, this is the small particle figure. Small particle values, where we see higher values in the higher latitudes where there's more productivity and generally larger phytoplankton community. The depth at where this kind of subsurface maximum appears are different between the different biomes, leading to different shapes of the curve. And it's also cool to see the data in this perspective because using this biome grouping, we are able to see different results between different biomes. We're not just averaging things together that maybe aren't so similar at this point. So now we can look at what these profiles look like for the larger particles. And this is a little bit of a noisier data set. But again, we are still able to see differences between some of the high latitude regions and the more low latitude oligotrophic biomes. In the high latitudes, we see more attenuation and less so in the lower latitudes. And so I think it's super interesting to look at these profiles, but it's easier to kind of make some comparisons when we collapse these profiles into one value and compare the transfer efficiencies of the small particles on the left and the large particles on the right. And so we can see when we're doing these comparisons by biome, we're seeing that the larger particles have a higher transfer efficiency than the smaller ones, which we would expect because the larger particles are likely heavier and therefore can sink through the water column faster, escaping remineralization and therefore transferring more carbon effectively or efficiently. And for the small particles, they're lighter so they can undergo more remineralization. We're also seeing this trend where at lower latitudes or the lighter shades, we're having higher transfer efficiencies than the higher latitudes. And so when we have these latitudinal trends, we can think about how does the transfer efficiency vary with temperature. And so here we're looking at the surface temperature, which we're defining as between 0 and the primary production zone depth in each biome. And we can see an increase in positive correlation between the transfer efficiency and temperature. This kind of result was found in Henson et al. 2012, where with increasing temperature, we're seeing an increase in transfer efficiency, but kind of sometimes more classically, we would expect, and other papers have found as well, that when we're increasing temperature, we would expect a decrease in transfer efficiency because there's higher rates of respiration with temperature. But we'll compare how it looks when we're looking at the depth values in a little bit, but just something to keep in mind. But also, we can compare how the transfer efficiency varies with other biogeochemical parameters. So we can look at the oxygen minimum, and we see that there is a negative correlation between the transfer efficiency and the minimum oxygen, which we're defining here as the 10th quantile to make it less sensitive to outliers in each biome. And so this kind of goes with what we would expect where when you have oxygen, it's easier to respire and consume these particles than when there's no oxygen. And we can also look at how it varies with nitrate. And there again, there's another negative correlation here, but we can kind of see that the green, which is representative of the Southern Ocean, which is a high nutrient, low chlorophyll regime, is kind of skewing this. And when we look closer, we kind of almost see a separation of two different clusters where we can think about maybe there are two regimes where one, we see this high transfer efficiency where there's not many surface nutrients, and the lower transfer efficiency when there's higher nutrients. And there's kind of this question that we can ask that was brought up in the Henson paper about how much nutrient recycling occurs and how this might impact the types of particles that are formed in the surface ocean. So that paper also looked at this metric called the biological carbon pump efficiency, which looks at what percentage of nitrate available is consumed. So in this, the nitrate available is looking at the nitrate in the 100 meters below the surface ocean. And so when we look at this figure on the right, if we're at closer to 100% efficiency, that means all of the nitrate available is consumed. So there's lots of recycling, versus the left, only a small fraction is consumed. And this kind of, we can see this kind of separation a little bit more clearly here, where maybe in these regions with low efficiency, we're producing more labile or tasty or particles that are more easily respired compared to a higher efficiency regime where we're making more refractory or less tasty particles. And that's why they're able to escape through the water column. And so as I mentioned before, these were just comparing relationships between the surface ocean parameters, but we're evaluating the transfer efficiency between the primary production zone and 500 meters. And we see that these relationships are not as strong as the surface ocean, which kind of brings up, again, the balance of the importance of the surface ecological community and the biogeochemistry versus the actual respiration rates that occur at depth. And just, we could do the same analysis for large particles, but just going to go through this very quickly, that they follow the same temperature, oxygen minimum, and nitrate trends as the small particles. And so what we've been looking at for most of this is the annual or the climatological average, but these floats are making measurements in all of these biomes year round, so we can look at annual cycle as well. And so for this, we're just going to look quickly at 3 different biomes, a high latitude biome in the North Atlantic in red, and the Southern Ocean in green, and then a more low latitude equatorial region in the North Pacific in light blue. And so there's a kind of a lot on this slide, so I'll walk through each figure, each part of this figure first. So in the upper left-hand corner is showing the primary production zone integrated particulate organic carbon, which is converted from the small particle backscatter signal. And we can see that in the North Atlantic in red and the Southern Ocean in green, we have periods of really high productivity where we're producing a lot of carbon in the surface ocean, and they're also out of phase because they're in opposite hemispheres. And then when we're looking at the North Pacific, we don't see much variation or really a signal at all from the small particles because there's less carbon in the surface ocean there. And then if we look at the upper rightmost figure, we're comparing the change in the primary production zone depth, so what we're using as that reference depth or considering the surface ocean. And for the North Atlantic, we see that there's more variation in the primary production zone depth compared to the Southern Ocean. And then we don't see much change in the North Pacific, which again is what we expect because there's not much seasonality in these lower latitude regions. And so kind of looking at these degrees of seasonality in these metrics, we can look as well at how the transfer efficiency of small and large particles varies with time. And what is quite striking, at least to me, for the North Atlantic is we can see a high degree of variability in the transfer efficiency, more so than the Southern Ocean, but also in both cases, we're seeing lower transfer efficiency during the periods of high productivity. And I think that we can continue this analysis comparing between multiple different biomes and kind of ask more questions. So there's many questions that you could ask here, but this is kind of where it's at this stage. So just to wrap things up and some conclusions and takeaways, I think this work is particularly cool because it's leveraging the whole float array to see larger patterns across both space and time. In general, we're seeing that larger particles have higher transfer efficiency than smaller particles, and we're also seeing higher transfer efficiencies in warmer regions. And this kind of begs the question of what matters most when we're thinking about the biological carbon pump, or where the balance is most important, of the types of particles formed in the surface, which are really influenced by the biogeochemistry, versus the remineralization rates, which are more temperature driven in the water column. And for that kind of analysis, we were looking at the climatological average, but it'll be interesting to see if those kinds of relationships hold true through that annual cycle or bloom period. Anyway, that is the end of my talk. Thank you for listening, and happy to take any questions.
Thank you, Ellen. I think we will hold questions until the end. If you have questions, please add it to the chat. So I just want to make sure we get through both talks. So yeah, if you have questions, hold them until the end or add it to the chat, and we'll come back to it. Thank you very much, Ellen. The next speaker is Adam Storer. All right. Thank you, Adam. You're still muted, by the way.
There we go. Can you hear me okay?
Yep.
Okay, great. I'll start. Yeah, thank you so much for having me today. I'll be talking about the work I did for my master's thesis with Dr. Katchett-Fennell, and that involves estimating marine phytoplankton biomass and production from the biogeochemical Argo float array. With BGC Argo, I mean, the big benefit to it is that we can get at subsurface biogeochemical measurements, especially measurements relevant to phytoplankton, that will basically really help our understanding of the subsurface ocean. And when it comes to describing like dynamics in phytoplankton biomass and production, what's often used is satellite data, and satellite data only contains information, or mainly contains information from that surface layer, the first optical depth. And so they don't contain that subsurface information about the deep chlorophyll maximum or deep biomass maximum. And so BGC Argo is that big advancement that we can use to better understand variations in phytoplankton biomass and productivity. And we go, on the right there, you can see that, common picture of satellite surface chlorophyll that's often used to describe phytoplankton dynamics. And so with the BGC Argo array, I mean, it's accumulated this massive database of high quality chlorophyll and particle backscattering measurements. And you can see just in the map here, I and several other colleagues there did just a census of how much high quality data there is. You can see that you have pretty darn good coverage of the global ocean with all these biooptical profiles from BGC Argo. And so that's the focus of my talk, is using this data to better describe that biomass and productivity of phytoplankton. And I'll be splitting my talk basically into two parts. And the first one being talking about a method for estimating depth resolved net primary production using these particle backscattering measurements from the floats. And then in part two, I'll talk about global scale depth resolved phytoplankton biomass dynamics using the Argo float array as well. So for part one, this is work that's been published in L&O Letters, and that's the estimate of net primary productivity. So that work is inspired from this wonderful paper from Ken Johnson and Mariana Biff. And what they did in that paper was effectively they extracted daily cycles of dissolved oxygen from the Argo float array. And I'll talk about in the next slide how they did that exactly. But basically, they get an average daily cycle of dissolved oxygen, which you can then infer based on the rate of change in oxygen throughout the day. You can infer how much gross oxygen production is going on, and then using an empirical ratio of gross oxygen production to net primary production, you can infer net primary productivity. And so in that paper, they estimate global net primary productivity to about, I think it was 52 petagrams per year. And that's pretty much in line with what satellite models give as a global total. And so this is really useful information here because we can get at that subsurface profile and production that otherwise we can't observe with satellites directly. So our question was simple, which was like, can we do the same thing with particle backscattering measurements from the BGC Argo network? And so what we did was follow very similar steps to what Johnson and Biff did. And that involved identifying floats that were profiling at non-integer intervals. And so what that means is that each successive profile that float takes is taken at a different hour of the day. And so if a float's profiling at 10.2 days, that non-integer interval, the first profile might be taken at 3 p.m. and the next one 10 days later is taken at 6 p.m. during the day, and then 9 p.m. after 10 days, and then so on, so that throughout the float's lifetime, you have measurements all throughout the day. And in this case here, you have a float that basically, in this histogram here, you have profiles that are roughly taken equally throughout the day over its lifetime of several years. And so we're interested in using that kind of float for calculating an average daily cycle. And then in this other example here, this float is just profiling every 10 days. So it's usually, it's almost always coming up at the same hour of the day. So that we don't want to use that kind of float for calculating an average daily cycle because we're going to have biases in our biogeochemical measurements at that particular hour of the day. So we want something that's equally distributed. So what we did is we applied that filter to the whole BGC Argo network. And what you see in these maps here are floats that are suitable for calculating an average or median daily cycle of particle backscatter or oxygen. And so just note here that we have different sample sizes between the backscatter floats with backscatter sensors and floats with oxygen sensors. And what we do here with this data is we calculate that average or median daily cycle of backscatter and oxygen for each latitude and also for seasons. So just in this show here, we have dial cycles at 5 meters depth, and you can see that carbon estimated from the particle backscattering sensor at the top row there increases during the day alongside oxygen, and then it declines at night when there's the lack of sunlight and respiration is dominating. So from those that variation we get throughout the day, we can fit a model to this, which basically smooths out a lot of this this noise, I guess you can call it, or biases, and we can get an estimate of gross carbon production and gross oxygen production from daily cycles taken at different latitudes and at different depths. And then we can go back and estimate net primary production from those values. So the bottom row there is what Johnson and Biff did, and then at the top row is we're doing a similar thing with particle backscatter. And so with Johnson and Biff's method, they get the gross oxygen production from those daily cycles of oxygen, and they divide that by that empirical ratio of 2.7 from Mara, and that gives them an estimate of net primary production. So we followed a really similar steps. We had add a couple steps here. So in our version, we had to multiply it by photosynthetic quotient to get an estimate of gross oxygen production. And then we also accounted for dissolved primary productivity down at the bottom there. And then we use that 2.7 value again. And so we have basically two independent estimates of net primary productivity from these floats. And so once we apply those steps to different depths and different latitudes, from those average daily cycles of carbon and oxygen, we can get a profile of MPP. There are differences between the two of them, like you can see here, but we should also note that they come from different samples, because not all floats have backscattering sensors on them. So we can see kind of the general trend of it declining with depth and approaching zero towards the euphotic depth. We can do this with by latitude. We can do it again with MPP by season, which is shown in this representation here, where you know, in the summer it increases and then declines during the winter, which is consistent with what we expect for the Southern Ocean, and also consistent with the seasonal variability that Johnson and Biff were able to detect in their paper as well, for a different region though. And so the main points of this is that we can get MPP, like depth resolved MPP on basin-wide scales, that's effectively working like an average of the last decade of measurements. And so that's really useful because, you know, if we were going to go out on a boat and take water samples, these aren't point measurements and there could be a noise. And so we're getting a decadal, a long-term average of MPP throughout the water column. And these estimates of MPP are comparable between the two, which you can see in the red and blue column there. When we add up all the latitudinal averages that we had in the previous slide, and those totals are within range of what satellite models give you from the Oregon State University website, from Barenfeld's group. And then with these depth resolved MPP profiles, we can help tune models because we have more information about what's going on below the surface. And we can also use that to monitor subsurface primary productivity. So that concludes the first part of my talk, and then I'm going to move into biomass now. And so global scale depth resolve phytoplankton biomass using the Argo array. And this is work also from my thesis, but in prep for submission. So like in that map I had before, we have a you know, pretty good coverage. What you see in that map there, we have high quality chlorophyll A and backscattering profiles following a very similar quality control to what BGC Argo already implements for chlorophyll A, and the algorithm suggested by Giorgio D'Almo and then colleagues. And so we have, you know, global scale measurements of phytoplankton throughout the euphotic zone that we can use to better understand phytoplankton dynamics in the ocean. And we can also compare that with that commonly made assumption of surface chlorophyll A acting as a proxy for carbon biomass, which is, you know, commonly done in satellite studies. And so I'll just quickly go over the methods here. I'm going to take some liberties that people are already aware of, you know, using backscatter as a proxy for carbon biomass. But we use the algorithm from Jason Graff and colleagues to convert particle backscattering to phytoplankton carbon. And then we use the chlorophyll estimates from the fluorometer on the floats. And so with all those profiles there, we calculate monthly and annual averages as climatologies for every 10 degrees latitude for the Pacific, Indian, and Atlantic Oceans. And so we basically, you know, have the Atlantic here, and we divvy it up into 10-degree slices of latitude and calculate a monthly climatology from those. And so, or no, not monthly, more like a weekly climatology. From those climatologies, we then try to answer two things. We estimate how much phytoplankton there are on Earth in terms of carbon, as well as the total stock of chlorophyll A in the ocean. And then the second goal for this was to compare the seasonal cycles in surface chlorophyll A with those of depth integrated carbon, effectively testing, you know, the the assumption that's commonly made where surface chlorophyll is a proxy for phytoplankton biomass. And so using BGC Argo, we can greatly constrain how much phytoplankton there are in the ocean to about 340 teragrams by adding up all those latitudinal averages and calculating the total stock within those areas. And if you compare that to previous estimates, which range from about 250 to 2,400 teragrams of carbon, that's a very, we're very constraining this number down to a much smaller range using BGC Argo. And part of that uncertainty that has come before was because we didn't really know what the ratio of chlorophyll to carbon was, and or it came from like shipboard measurements, which are relatively sparse, and there's more uncertainties involved with that. And then if you compare this to the most recent estimate from Barn and colleagues, they give a best estimate of about 600 teragrams. And so that's roughly, double what we have from BGC Argo. And the big value of this is that we have that subsurface information too, so we can account for maxima and chlorophyll and carbon that occur, below the surface. And you can see that here kind of towards the oligotrophic regions of the ocean. So we get this constraint on global phytoplankton stocks. We can also use the BGC Argo array to better understand seasonal patterns between surface chlorophyll and depth integrated carbon, as well as comparing the two. And what you see here is just the how the total stock of carbon and chlorophyll varies throughout the year by week. And it's all made relative to the summer solstice. And so it's aligned with, the day where we have the most sunlight. And that's needed to sort of makes things, fit nicely and work within the seasonal solar cycle. So from those seasonal cycles, we can then calculate bloom metrics. And so one of them is peak timing, which probably a lot more people are familiar with. And that's just what week of the year relative to midsummer do we see the most amount of surface chlorophyll and the most amount of depth integrated carbon biomass. And that's just shown by these stars here, right? And then we can do the same thing, which bloom duration, which is just the weeks, the total number of weeks above the annual average surface chlorophyll or depth integrated carbon. And then we can do, we also have another metric, which is called bloom amplitude. And that's the range of surface chlorophyll and depth integrated carbon you see throughout the seasonal cycle divided by its annual average. And so that's sort of like a coefficient of variation that's useful for understanding, like comparing the variation between the two, right? So we have these three bloom metrics, right? We calculate them for each latitude that we have there. And then we can compare those bloom metrics. And, these are, somewhat arbitrary choices, how we calculate these and what they're called. But the point is that they're useful metrics that allow us to compare them quickly. Although the differences are, you can, see obvious differences between the two just in this figure here. So right here, these are these bloom metrics I just talked about. And what you can see here is that with this first panel on the left, peak timing and surface chlorophyll is very consistently before that of the peak timing of depth integrated carbon biomass. And so what that means is that when you see a peak in surface chlorophyll in the water, it more often than not means that that's not the peak in phytoplankton carbon, at least in terms of the seasonal cycle. We can also see that with bloom duration here, this middle panel here, blooms that tend to last longer, at least relative to their annual average. There's different definitions of bloom duration, but the point here is that we don't get similar patterns here. And then in this final figure here, we have bloom amplitude, which is that range divided by the average, effectively a coefficient of variation. And you can see here that in the lithotrophic gyres, you get far more variation relative to their annual mean in surface chlorophyll than what you get with depth integrated carbon. So there's less variation with these blooms. So if you're using surface chlorophyll as a proxy for phytoplankton biomass, what surface chlorophyll tends to give you is blooms that occur earlier than what actually happens. They tend to look shorter, and they tend to have more variation than what's actually happening in the water column based on these BGC Argo measurements. So in other words, they don't serve as a very good proxy for phytoplankton carbon biomass. And that's not entirely a surprising result, but there's a lot of good work that's been done talking about these chlorophyll to carbon ratios. But it's an important thing to show with this BGC Argo database, because effectively we can monitor depth resolved phytoplankton carbon at a global scale now. And this will help improve our understanding of phytoplankton dynamics. And so I'll finish up the main takeaways for these two parts. The first being is that we can get depth resolved net primary productivity from diel cycles of particle backscattering. And that's from the work published in LNO Letters here with Katja Fennel. And then for the second part of the talk, BGC Argo has grown and advanced to a point where we can get at global scale depth resolved phytoplankton biomass dynamics, which is a very important advancement in terms of understanding blooms and what triggers them. So thank you for having me. I'll let everyone move to question period now. Thank you.
Thank you, Adam. Thank you, Ellen, for the two excellent talks. We have a few minutes for questions, and then so there has been a pretty lively discussion for questions and answers for Ellen's talk, and so I'm gonna try to avoid questions that were already answered in the chat, but there is one. So this is from Ken Johnson. Adam, any hope of getting diel vertical migration cycles from diel cycles of BBP spikes?
So yeah, I think that's probably possible. I think Nathan Briggs has done something sort of similar. Like he's pulled out backscattering spikes relevant to the solar cycle. But I'd have to look at it more carefully. But I think that's definitely possible.
Great. Another quick, this was answered, but I would kind of like to hear a little bit more about this. So this is from Yang Zhang. Great talk, very exciting data set. I wonder why the subtropical gyres tend to have a higher transfer efficiency where there are more small particles. Your results show that smaller ones tend to have a lower transfer efficiency than large particles. Do you have any comments on this inconsistency? And I kind of had a similar thought too, because I would assume that respiration rates, because respiration rates are higher at higher temperatures, you would expect an opposite relationship, so things get, respired faster in warmer waters. And work by Peter Brewer, for example, showed very strong correlation between temperature and AOU from cruise data. So anyways, I just, you answered it, but Ellen, if you have any further thoughts on that, that'd be great.
Yeah, I think this is something that I've been thinking through a lot about what influences it, but I think kind of in this case, at least doing it this way compared to sometimes more of the sediment trap approaches, you're seeing that the types of particles that are formed in the surface ocean might play a more prominent role compared to the respiration rates if it's not desirable material. I guess that was kind of the main point or thing that I've been thinking about the most recently.
So you think it's just more of the material, the characteristics of the material that's produced rather than the temperature effects. Great. So the next question is for Adam, for Guillaume. Thanks, Adam. Awesome work. You mentioned the bloom duration. How did you define the start and termination of the bloom to calculate your duration? Did you use a specific method?
So the method I used was it's not the termination initiation. I didn't use those definitions. I used basically, well, what I used was that it's the number of weeks above the average surface chlorophyll A or depth integrated carbon, and that's based off of what Daniel Boyd, Daniel Boyce, sorry, did in a paper. So it's using like I think I know what you're talking about with the start and termination. It kind of gets, if you use those definitions, it starts to get funny, because if you have like a second bloom, like a secondary bloom, the metrics like between carbon and chlorophyll start getting funny, because maybe that secondary bloom has a much higher growth than the first bloom, and then it's not the same story with the carbon. So I tried to use metrics that were more fitting for a possible secondary bloom throughout the year, if that helps.
Got it. Thank you. Great. This question is for Ellen. This by Ken Johnson. Any sense that you can derive sinking rates from the data by following pulses to depth in each biome? There was some discussion in the chat, so if there was a big like bloom termination event where all, there's a big export event, you could maybe track it. People have done this by rapidly cycling floats or gliders in the past, but that's not in the near future, not really a possibility for a growing global array. And so I think Nathan Briggs maybe, he did a similar analysis for the fragmentation. I can't remember if sinking rates were quantified for that, but Ellen, if you have any further thoughts on that, that'd be great.
Yeah, they were quantified in that paper. This has been on my to-do list, and I like went at a first stab, but shifted a little bit. But that's definitely been something that I've been thinking about, of maybe you can use floats in a similar region, because I think when you're taking the biome approach, it's such a large region that you could have blooms that start at different times within a biome. So I feel like you might have to group floats that are similar and then hope that they're close enough and are seeing the same things, but it requires a bit more work, which is on the list, because it would be great to have the transfer efficiencies and the sinking rates.
And do you have any intuition or like knowledge about, if the sinking rates are the same, like mid-bloom versus termination of a bloom, do different, right? I don't know.
Yeah, I think that's a great point and super interesting to think about, because I think that would also go back to what kinds of particles are being formed and when they're heavier or lighter. I would imagine changes based on the ecosystem composition.
Okay, great. Okay, last question. These are fantastic questions. Thank you to the audience for submitting these. This is from Lionel, and this is kind of related to bloom dynamics. Isn't the decrease in the nighttime POC heavily affected by grazing, and how does that affect the MPP estimates?
It would affect the MPP estimates. And so the assumption is that community respiration is equal throughout the day, and with dial vertical migration, I mean, this is that's going to lead to higher respiration at night than you have during the day. So I'd have to think about how that affects the MPP estimates. That's a good question.
Great. And there are other questions in the chat that we didn't have time to get to. It's 11 o'clock here now, and then so this is the end of the webinar. But Heather, I just want to verify that the chat will get saved somehow and we can get access to it. So maybe Adam and Ellen, you can respond to the questions maybe over email or something like that.
Yes, everything will be saved, and I will send the chat file directly to Adam and Ellen. Thank you.
Great. And this talk should be archived on the OCB website and also the GO-BGC YouTube channel, so I don't know how long, maybe a couple of weeks it'll be up. And then, so please spread the word, these are fantastic talks. The more people that see it, the better. So thank you everyone for attending the webinar, and big thanks to Adam and Ellen again for the two fantastic talks. They're very cool. So,
Thank you for having me.
yeah, great.
Everyone.